Hello? Hello? Oh. We had another person. Okay. There we go. So how's everyone doing? Oh,
Great.
Sushmanth, you're muted, or your audio's not working. Okay. Oh, hello. So you're doing good.
Yeah,
All right.
I mean, I'm a little nervous, but other than that,
Yeah. But Shalini, how are you?
Am I audible?
Yes, you are. Hello.
Yeah. Hi.
Hi.
Last 1 week I was like pretty bad with health issues. I was affected with dengue actually.
Oh,
I couldn't work properly. That's what all was going on. Actually, Bradley, I want to give a small presentation about the proposal thing which I mentioned.
okay.
Can I give it today?
Yeah, you can go ahead. Yeah.
Okay. Can I start or should I wait for Gordon also?
We can start now.
Yeah. Okay, cool. Thanks. My screen is sharing, right?
Yes.
Yeah. Actually, after there are some points I need to mention before explaining. DevoNet is completely new, actually, Bradley. I mean, this idea is never implemented in DevoWorm GSoC project at all.
Okay.
Because whatever segmentation we've done till now is a semantic segmentation, actually.
Okay.
I mean, this really means like every cell is classified into one class. I mean, all cells will be like, but it is not good at all because semantic segmentation generally happens for tissue level. If you want to segment tissues, it will be like good idea to implement semantic segmentation. But for cell level, we need to implement instance segmentation, and it is completely related to GNN what we are actually applying because can you see here, Bradley? This is like stage 1 of DevoGraph.
Yeah.
Implemented by Jiang Li, and you can see here he mentioned the model works well for images where cells are sparsely populate, but not for cells densely populate. I have checked actually. Not only I have checked up till 32 cell stage, DevoLen is working pretty good. But after 32, I increase it 10 cells. 10 cells. It is not working properly. That's the main issue. I mean, we can create a graph neural network up to 32 cell stage, but at the end, while we are extracting the CSV file is there, right?
Yeah. Yeah.
Whatever in stage 1, we are extracting a CE raw data CSV file. And this is not good for after 32 cell stage. I mean, can you see the cell name were actually not predicted actually correctly, and the time and the points also when you plot on a 3D graph, they are at different points, which is not related. And here comes like, you told me to refer GSoC idea of 2019, 18, 17, right? I have gone through them. Okay. This is DevoLen output actually. This is from the stage 1, this picture from stage 1 itself.
Okay.
At 232 frame, it is not working properly. And here comes the DevoNet. This is the segmented image, not original image. This is segmented image, which I have segmented by myself. I applied for it. See, I mean, can you see these results actually? See, here centroids were not extracted properly. Cells are not even segmented properly.
Yeah.
But here, every cell is segmented properly, yeah. And I have gone to 2019 GSoC results also. I think, like, see, here the picture of 3 cell state, it is not segmented properly, and cells, I mean, we can't extract the centroids of this. Even DevoLen works properly than 2019. And I have gone through 2018 GSoC, and this is like some lineage population. I didn't got what it is. I need to go on, and I have went to 2017 segmentation results. Yeah. This is 2017 segmentation results. See, I mean, here also the cells were not segmented properly. We can't extract. And 2017 GSoC idea mainly concentrates on images, not on video data.
Yeah.
Yeah, I mean, we can't implement. And this data is like 2D data set. Actually, right now we are working with 3D data set, like pointing out and all this. So it is not a good idea. So, and you told like our whatever is there, like idea list mentioned here. Sorry, this is like, sorry, 1 minute. Can I just 1 minute? Yeah. Actually, you mentioned here in the first idea, first work, refining means to segment raw data and incorporate into DevoGraph pipeline.
Yeah.
And you mentioned will require refactoring CNN models and understanding biological training data sets. So, see, I mean, I had tried to improve DevoLearn segmentation. But what happening was DevoLearn is completely built on a machine learning algorithm, Resonate 18 structure. So we can't segment it properly using DevoLearn. We need to implement. Do you know unit structure? It is mainly used for biological data. Actually, it works with biological data.
How do you spell
that is
it?
doing
Unit structure.
Yeah, unit structure.
Okay, I'm not really familiar with it, but yeah.
Okay. Unit is working pretty good actually. I have gone through, these are segmentation results. I mean, at 320 cell stage, where it is, sorry. Okay, at 320 cell state, the segmentation you can see, like every cell is like segmented properly. When I'm trying, I just need to complete work with that extracting CSV file. That's, I mean, I can't work this due to health issues. I was not able to work properly.
Yeah.
But if we try to extract centroids, and another thing also, in DevoGraph stage 2, Jiangli hasn't used the CSV file which he was getting in stage 1. See, this CSV file is CE raw data CSV, right?
Yeah.
And in stage 2, as an input file, he's not using that. He's using some different data file. I mean, CE raw data 1 year part CSV. I don't know why. I mean, we need to give input of DevoLearn stage 1 output to the DevoGraph stage 2 actually. But he brought somewhere this file. I don't know where he got. Bradly, seriously, it took these many days to understand the complete structure of DevoGraph underlying. Okay. Actually, Gautam also saw this thing. He's using some different CSV file. He actually shifted to another ML project. I mean, he thought like it can't be done. But see, through my proposal, we can, I mean, he also mentioned like we can extract cell volume and here. Moreover, the simple input file in this notebook contains the column called cell, which contains the volume of cells observed at different timestamps. By my model DevoNet, we can extract these 3D data points at different time frames and this size of each cell also. You got it, right? I mean,
yeah.
I mean, it is completely related to graph neural network only. Actually, we are completely working on stage 1 of DevoGraph. Clearly, we are incorporating DevoGraph stage 1 pipeline to stage 2. Actually, Jiangli worked pretty good at this one, stage 2. We created this node embeddings using KNN. But the stage 1 is little messed up actually. He hasn't given the CSV file which we are getting from the stage 1. He has given some other CSV file. I mean, C raw data CSV, and here the input was different. I mean, raw data, there are 2 different files, and actually we can't extract size of each volume of each cell from DevoLearn. DevoLearn is just for segmenting and extracting 3D points, right? Yeah. But how can he got this size of each cell? So we can't implement DevoLearn actually, or even 2019, 18, 17 model.
I think for last year's project, we used a couple of different data sets. So we have a data set where it's numeric only, and then we have a data set where, we have the data sets that are images, and so I mean there are different ways that you can train this. See, we work on C. elegans, which is where you have movies and you have images, but we also have things that are like other types of data that we've like extracted, and the cells have names, so there's a nomenclature and a lineage tree. So we have all these things that we can train the model with. So that's where I think some of this stuff comes from that is a little confusing. But I think this is a great. This is a great.
You got understood, right? What I really wanted to do.
Yeah, no, I do. Yeah, it's great. Yeah, it looks great. I mean, it's
Can we implement?
What was that?
Actually, we can extend this to a paper also, research paper.
Yeah.
I mean, this method is not implemented in C. elegans actually, instance segmentation till now. So we can implement and we can extract the points, even cell area, cell volume, and whatever data we needed, we can use it to DevoGraph stage 2, and we can keep this DevoNet model as a different one.
Yeah, I think so.
That's what from my side. I mean, right now it is like you understood, right? It is completely related to.
Oh yeah, yeah, definitely. Yeah, the way because we now if you want to use like other types of training data, we have them, but I don't know. This may actually be good for just kind of using just a lot of video data or image data. Yeah.
Actually, DevoWorm was built on this data, cell tracking challenge data set, and I was using the same data set actually. So, I mean, we can't try with different models. We will use that data and extract CSV file, whatever we want, and we can incorporate into DevoGraph stage 2.
Yeah.
Okay, I mean, this idea is clear, right? I mean, this is related to GN only, like the comment you have mentioned.
Yeah, it is, definitely. Yeah. So, I mean, in your proposal, you'll want to mention this, like, you'll want to kind of build up.
Sorry, I think there is a mistake. What? Actually, Mayuk and Mainak were analyzing my idea, and they removed some of the points. I need to work on this. I will keep the proposal once again.
Yeah. I mean, that's why I like to see the proposal first. Like, you get I usually go over proposals with people, and so anyone who's applying to GSoC, I can look at your proposal before you. And I recommend that you let me look at it before you turn it in, because it's always like, it's always good to go over the different, especially the schedule and the description and everything. Now, Sushmanth, I think you did a pretty good job of describing the problem and in general, and then the schedule I think is laid out pretty well. I'd go over it again and make sure, like I think I mentioned before. You want to make sure that you've built in redundancies for if something doesn't work, if you can't implement something, what are you going to do for that week? You don't want to sit on things.
Actually, what I did was, Actually, I implemented this thing with mouse embryo cells. I haven't worked on C. elegans, but I trained a model on mouse embryo cells, and I just segmented that C. elegans data. I mean, then I got those results. I mean, this will work. Actually, I mean, I have the results. I have wrote the code also, but I need to extract this .py file and the volume and cell names.
Right.
Okay. I will mention it clearly in the proposal of next round of review. That's it from my side. Thanks, Bradly.
Oh, well, thanks. Yeah. Well, that's a great presentation. So you said you used mouse cells for some of the.
Yeah. I just did a parallel project to understand instance segmentation actually.
Oh, okay. Yeah.
Then I implemented the same thing for the C. elegans. So.
Oh, okay.
It worked pretty well.
Yeah. So I think we should. I like the idea of using a different organism just to get the feel for the cells and how they segment, because you have different in mouse. Of course, it's a different type of embryo than C. elegans. You have a lot of cell proliferation, and it's not as C. elegans has a very specific way that it divides. Every time you get this early polarization, you get other things that are going on. With mouse embryos, a little bit different, but it would be. I think that's good that you can pick out. You're making the distinction between instance and semantic segmentation.
Actually, I have tried with the deep Milligan star also. Those segmentation also pretty good. I mean, we can replace the whole DevoWorm with this thing. I mean, DevoWorm have the cell segmentation, right? We can replace cell segmentation of DevoWorm with this DevoNet. So I will change the proposal and I will let you know with clear documentation. So that would be okay, I think.
Yeah, that's good. Yeah. Well, thank you. Yeah, that was a good presentation on your progress so far. I look forward to seeing it. Yeah. Yeah. Okay. Well, that's a lot of work too. I thought.
I would have implemented, showed you the model also. I was having health issues, so I couldn't work.
Oh no, yeah.
And by next week I will show you.
Oh, that's fine. Yeah, yeah. All right. So, Dick, how are you?
Okay.
Yeah. Yeah. I know Susan's not going to be here today because she's at APS.
Oh, right.
Yeah. And we have a couple new people here. Or, well, Shalini is here. They're new. And Kritika is. Did you have any updates for us?
Just waiting for Bashir to approve the last version of the enhanced ensemble manuscript.
Oh, okay. He passed it off to him. Okay.
He's moved to an apartment, so things are very flexible.
Oh yeah. All right. Kritika, did you have any updates or?
Not yet, but I think I'll have proposals to. I'll send you. I'm fairly new to the format of it, so I'm quite confused, and I haven't made any contributions to DevoWorm, so I think I have to get into it more. So
yeah, well, that's yeah. It's definitely, you just have to look at what's there. If you need any information, I can send it to you. I still didn't send Sushmanth anything on the topological data analysis, but I'll get that out. I've been meaning to do that.
Yeah, Bradley, I'm not getting any resources recording topological data analysis on biological images or microscopy images.
Oh, okay.
I've been searching since around 2-3 weeks. I couldn't get one resource. Those all are whatever we have. It is completely different topological data analysis. I can't implement on microscopy images.
Right. Yeah, I mean it's not. I don't know how much work people have actually done on that. So it's kind of one of those things where it's I have to go dig up the things that I passed around last year, but I don't know how useful that'll be. So yeah. I've been keeping, just if you're interested in, if you're interested in learning more about what we're doing in the different projects. So this summer's project is built upon several years of projects that we've done in the organization. This is the DevoWorm repository, of course. Actually, it's the DevoWorm organization, and we have the DevoWorm repository, which was actually built in 2020 and 2021. And this is something, if you want to get familiar with what we're doing in the organization, you can go through the materials here. We have DevoWorm, we have DevoGraph, which is here. And then, those are sort of there. There's documentation. There are also a couple of different toolboxes that Sushmanth has pulled out, and we're making new repositories for. So we have the nucleus segmentor, we have the membrane segmentor, and the lineage population. And these are all based on the sort of semantic segmentation techniques that we're using currently in DevoWorm, although if Sushmanth is successful this summer, we might get some instant segmentation. We also have a couple other things in this organization. They're not really that important for the GSoC project, but we also have this DevoWorm. Let me see if I can go to the DevoWorm. So this is actually our organization, sort of an older set of repositories that we have. These are not focused on DevoLearn specifically, but these are just like all the stuff that we've done since the beginning of the project, or the beginning of the group, when the group started back in 2014. So in 2014, we started out, and we've been working on a lot of projects since then. We have a lot of repositories in here. The ones that are relevant to people working on GSoC are things labeled GSoC. So we have GSoC, GSoC 2022 is not necessarily relevant. Well, I guess we did stuff in 22 as well. But GSoC 2019 is things that aren't in the DevoLearn organization. I think there's a GSoC 2018 and a GSoC 2017, and they just go back a ways. You have to kind of go through this to look for it. But yeah, so 2018 is here and 2017 is here. And so if you go back to those projects, you'll be able to see what people were doing back then. We've done a lot of different things. We've done things with trying to predict lineage trees. We've done things with image segmentation, and we've done other kinds of projects. So those things may be useful to you. They may not be useful to you, depending on what you want to do, what your proposal's about. So anything from like 2020, 2021, 2022, those are all in the DevoLearn organization. But before that, we have things in this repository, github.com/DevoWorm. So that, I want to disclose the history of that in case you find that useful. The reason I want to do that is because we sometimes forget where we've been with things, and I don't want to reinvent the wheel, basically. So, those are things that you might find useful. So, any questions? All right. I'm going to go on to some other things here. Share my screen again. So I was able finally to get something together for what they call Darwin Day, which is on February 14th. And I've been meaning to post a blog post. It's usually something to do with evolution and evolutionary biology. And I've done a lot of different posts over the years since I started this blog in 2008, or actually 2009. And I've done a post, I think, since 2012, almost every year. So this was an opportunity to kind of revisit some of the themes that we've talked about in the group here in the past. And one of those themes I noticed by going over some of the things that we've done in like maybe the last year was we've had a couple discussions about ancient embryogenesis, not really the origins of life, but like the origins of embryos. And what was that?
Embryos are no more than 500 million years old.
Yeah. So yeah, this is what this blog post talks about. So the origin of life, of course, is much older than the origin of embryos. And if you go down to the bottom, you'll see that I have this tree. This tree kind of shows how the origin of life relates to the origin of embryos. So you have this last universal common ancestor at about 4 billion years. And you get these two different things called oxygenation events, which are where the Earth's atmosphere increased its oxygen content. So you have the great oxygenation event and then the second oxygenation event. And the second oxygenation event seems to be the thing that really kind of pushed us towards an embryo of some type. So, when we get to the last eukaryotic common ancestor, which is the last ancestor of all eukaryotes, which are what they call protists, fungi, plants, and animals, we don't really have embryos yet. We go through this second oxygenation event. We go through the advent of photosynthesis, and we end up with this sort of gray area where we don't know. We haven't seen much in the fossil record, but we do have these pieces of evidence, like from the Dashanto assemblage in China. There are some other examples of multicellular organisms, maybe a bit earlier than that. And so all these things are, yeah.
Wasn't there some literature questioning whether those Chinese things looked like embryos were really embryos?
Yeah, that's the thing I get into in the post, that there's there's evidence, like, they have evidence, they've written it up, and they say, well, this looks like an embryo. But actually what they're looking at is you have these different things from the fossil record that sort of have these properties of an embryo, but they don't have all the properties. So one example is this Bicellum brassieri, which is this billion-year-old protist. So this is like going back about 1 billion years. And these aren't really embryos. I mean, they kind of maybe look like embryos. But what they're doing is they have these different stages, and they have these different stages of differentiation. So you go from what they call a naked stage to something that's exhibiting differentiation and cell elongation. And then you start to get this cyst, which actually kind of looks like a modern mammalian embryo with an inner cell mass. So
Bradly, that the naked stage actually looks like some bacteria.
Yeah.
There's some bacteria that form colonies that look like that.
Yeah. Well, at 1 billion years old, it's kind of hazy as to the difference, right?
Yeah. I'm saying that stage might even be a prokaryotic stage.
Yeah, it could be. And so this is the kind of thing they have. It's not, we can't really say it's an embryo because it doesn't exhibit all of the properties of a modern embryo, but it's starting to get from this split between bacteria and eukaryotes, which is, it's back here somewhere, but the last, last eukaryotic common ancestor is here. It probably retains a lot of bacterial properties back in this time period. So, you're starting to get things that look like unique to maybe the modern type of embryo that we see now. And then there's also Cavosphera, which has developmental stages, but these aren't really the developmental stages that we see in any extant embryo necessarily.
Bradly, could you please put the URL for this in the chat?
Yeah, me. This. All right, there we go. Yeah. And so this is the Cavosphera. This shows the relationship. We have the last metazoan ancestor back here, and we have so it's in that sort of phylogeny, but you have these different stages that kind of look maybe like it's going through development, but it doesn't necessarily look like a modern embryo. Then the Dashanto embryos are, I think this is a picture of one of the Dashanto embryos. So, I mean, this is again, you don't have a lot of the things that you associate with a modern embryo. There are cell migration, there are, differences. You have these cells living in association. They're in this sort of massed environment. So you have, basically an egg. So if you ask the question, what came first, the chicken or the egg? Definitely the egg. And this goes back to this time. If you go back to the Cambrian, which is about 550 million years ago or so, and this is the Cambrian explosion that people talk about when they talk about the explosion of different types of animals that exist, you begin to see sort of the origins of eggs, you begin to see the origins of egg-laying behaviors. So they found, for example, fossilized burrows, and this is about 70 to 80 million years before the Cambrian explosion. You see, like, where they basically have interpreted these burrows as places where the animals of the time would burrow and bury their eggs.
Bradley, when did life, so to speak, crawling out of the sea start?
I can't remember.
Okay.
Yeah.
When you say burrows, do you mean marine burrows?
Probably marine burrows, yeah.
Burrows or,
yeah.
Or terrestrial?
I think like aquatic burrows or marine burrows, like some of the modern day organisms that, bury their eggs in the sand and, in the shallow.
Underwater.
Yeah. Yeah, underwater. So these are pancrustacea arthropod species, basically. So these are not really, these are kind of within the Cambrian explosion, but it's kind of coming out of that period and just before that period. So actually, what I found an interesting paper on this. By the end of the Cambrian explosion, you start to get different life history trade-offs related to clutch size, which means that these organisms are you can see differences in terms of the number of eggs that they lay and their length of time that they're spending, like before they lay their eggs, and the amount of time they spend laying eggs. So there's an interesting sort of already an adaptation related to egg laying there. And so basically the evidence, though, too, is where you get these well-preserved multicellular structures. So, you're starting to get these geometric structures that are multicellular. They're starting to form these little colonies, and they're not really reminiscent of any of the species at the time. So they can't link these embryos to any one species. They can't say that this is an embryo of species X. They can only say that this is an embryo or an embryo-like thing. And that happens a lot in paleo, like in paleobotany, for example. You'll find different. What's that?
It still happens. I used to study fish larvae. It was the same problem.
Yeah.
This larva of fish, the larva of some adult fish that we know.
Yeah, yeah.
Yeah.
Yeah. In paleobotany. Oh, yeah.
Okay, stupid question.
Oh, yeah.
I was involved with an attempt to try to do 3D structures of those. But I didn't have an X-ray machine. You need X-rays to get through structures like that. I think that work was done later. Do you have any serial sections of the so-called embryos produced by X-ray imaging techniques? Because they might be interesting for segmentation and stuff like that.
Yeah, I can't remember. I think I saw some in the references. There may be some examples. I can't remember which ones they were.
There was a guy named, let's see, it's your list. No, it's not alphabetized. There was a Swedish guy. His name started with a B, but I don't know.
Oh, yeah.
Trying to see if it's in your reference list.
Yeah.
I don't see it.
Oh, okay. Yeah. Yeah, I don't know. It makes me think that there maybe there is something out there.
Yeah, I'm just saying there might be some data sets that you guys could work on for segmentation.
Yeah, that would be kind of interesting. That would definitely be interesting. So, like in paleobotany, they have this problem where they have different parts of the tree or different parts of the plant. It's like the root system, the leaves, the trunk, and they're all different species because they can't say for sure that this belongs to species X. So that's really interesting. But, what we're basically trying to do is we're trying to find the place where on the tree of life we get the first embryos. And they're, so we still don't know. After all this is a lot of images, but we still don't know exactly where it happened. We know that there are a couple of different traits, though, that might define that sort of arise in embryogenesis, or that may be related to embryogenesis, that have to sort of, things that happen in these organisms that require an embryo, perhaps. And
Okay,
so,
I'm curious, are there any of these that have been found that go back to the age of the Ediacara?
I think, some of the, I can't remember. There was a discussion on that in some of the papers, and I can't remember too much about it.
Okay.
Yeah. Anyways, so there are a couple of things that we see in some of these early organisms that suggest that there's this need for an embryo. We have multicellularity, we have cell type and tissue differentiation, so we're starting to get different types of tissues emerge. We get axial polarity in the organisms. We get immune systems, things where you're needing to fight off bacterial infections and viruses. And then apoptosis, which of course we've talked about before, is this programmed cell death that usually accompanies developmental sort of shaping or sculpting of the phenotype. And so all those things kind of emerge during the time of some of these, the Cambrian explosion, basically. And they're putting this in this phylogeny to show where these organisms. So you go from like a circular organism that has a sort of radial symmetry, and then this is where you're starting to go down the bilaterian into the bilaterian clade. So a lot of the examples from the literature talk about bilaterian embryos. Now there are also early radial symmetrical embryos. That is a different literature that I don't really focus on in this post, but they do exist, and that's kind of it's its own little area. Most of those I think are from China, but you can take a look at that literature. I cite a couple of them in the bibliography. But this is where you're starting to get this bilateral symmetry, this left-right symmetry, and from that you start to get a nervous system and musculature. Interestingly, these radial symmetrical embryos, these early radial symmetrical embryos, they go down a different evolutionary path, and they tend towards these nerve nets instead of a central nervous system. So like, today's hydra, if you're familiar with the marine organism hydra, they have what they call a nerve net, which is where the nervous system is distributed across the skin. And that's actually a direct relationship to the difference between the triploblastic and the dipoblastic origins of tissues. So you have this 3-layered embryo versus the 2-layered embryo. And
that raises an interesting question. There's some hydra which form hydroid colonies.
Yeah.
And hydroid colonies can have hundreds of individual hydra in them, and they're connected by some sort of stolon. Is there any knowledge as to whether or not the nerve net crosses the stolon?
I don't know. I don't know enough about that.
In other words, are they brains?
Oh, well, I think they're they're brains, but they're not centralized. They do a lot of the things that brains do.
Just raising the question whether they have distributed knowledge.
Yeah. Well, it's just in the different parts of the body. Like, it's basically arises from the skin. So, like, So it's coterminous with the skin, and wherever the skin is, it's those are hydroid colonies. So that's interesting. And then from down the bilaterian lineage, you get things like a through gut, which means you have a mouth and an anus and a gut, and then you get limited body regionalization. So you get start to get different structures like muscle pads and other things like a heart or some other organs that are within the maybe in some region of the body that's different from other regions. And then you also get segmentation. So you get these different in invertebrates. We see a lot of segmentation, and that relates to Hox genes. And they talk about the origins of Hox genes as one of the things that they're using to sort of interpret some of these embryos. So you start to see segmentation start to form, and in say like Acaria, and you can link that to sort of the beginnings of Hox, some of the Hox gene evolution that goes on. So it's interesting that you can take the molecular data and map it to what's going on in these embryos. You also get the central nervous system. You get a discrete head and other segments and appendages.
The fossilized so-called embryo is actually measured Hox genes.
No. Yeah. Well, yeah, we don't know that for sure, but
this is a correlation.
Yeah. So I talk about the fossil evidence. I talk about some of the things with going on with some of the molecular information. And again, the molecular information is something that you can take and you can look at like divergence times between different genes. So you can, look at the genes in one organism versus another, and you can do an analysis of their divergence, and you can pin their divergence time back to a certain point. You can look at gene duplications, or you can look at like the origin of a gene, and you can time that as well through mutations and other types of measures. And then you can reconstruct a tree like this. So that's what they're doing with the molecular data. It's just from modern organisms mostly, and they're able to infer a lot of these things back in time.
Okay, so I see a discrepancy here.
Yeah.
If you take an extant animal, or plant, or whatever, and you look at Hox genes, when do the Hox genes kick in compared to the blastula stages of embryos, which if these things are embryos, they would be similar to?
Yeah. You mean when they get expressed in development?
Yeah, when are they expressed compared to the presumed stage of these presumed fossil embryos?
Yeah, I don't know if they know that one. Yeah, it's just kind of like they're looking at these things that are emerging, but they don't.
Yeah, my impression is Hox genes are fairly late.
Yeah, I think so. And there's no fossil evidence that shows any of the later stages of development that I know of. It's just showing kind of early stage embryogenesis.
Oh, except for you had one picture of a dinosaur about to hatch.
Oh yeah, well that's a different period of time. Some of the other things I have in here are, talking about the molecular evidence, and then talking about the emergence of nervous systems and brains, which I always find interesting because there's a kind of it's similarly hand wavy in some of the things, but we know that synaptic proteins have evolved very early in sponges, and they've been around longer than brains. So it's interesting how those things have kind of gotten put in place. But the other thing is that you have these remains of once you get further down the line in evolution, you get towards the Cretaceous period, you start to find complex organisms in eggs. And this is an example of an embryo, or like a almost hatched dinosaur from, I think, the Cretaceous. And this is showing the egg of a dinosaur. The dinosaur is in the egg and it's ready to hatch. And you can see that it's kind of folded up, the legs are folded up, the arms. And so they were looking at the different postures of this dinosaur, and how the body folds up inside the egg. And this is kind of the first example of a really complex organism inside an egg. I know it seems like it's kind of late in evolution for that demonstration to be there, but it's a fossil record, so it's hard to find things that are in embryos, because embryos are a lot of soft tissue that doesn't get preserved. So
on that question, has anyone attempted to look at organic matter in the so-called embryos that are fossilized?
I don't. You mean like stuff that's remaining, like maybe potential soft tissue?
Yeah, lipids, for example, will often preserve for a long time.
Yeah, I'm not. I don't think so. I haven't gotten into the literature that deeply, but
Okay.
Yeah.
Actually, how, a small doubt is there, a stupid doubt. I mean, how can we extract this X? I mean, are they really available as fossils? Because when a living organism dies, right? I mean, how can we extract a egg organism inside an egg? This may be a stupid question. These are dinosaurs embryos, right?
Yeah, well, they have, like, these things get preserved in different ways. So there's a lot of preservation. Sometimes if there's a volcano, or if there's like some sort of lava flow or something, you'll get like conditions where things can be preserved quickly, or sometimes they get preserved in different conditions where there's no oxygen, so it gets, it's just the right set of conditions for it to be preserved and not, doesn't disintegrate. So sometimes you get those kind of conditions. A lot of the ancient DNA is actually from long bones and other types of bones. So there's a bone matrix that, gets preserved. If it gets preserved quickly enough, you'll have, tissue inside that's where you can get DNA out of. And there are other biochemicals where that's also the case, where you can get things from fossilized remains. And fossilization is a complex process. So you get like, the bone typically gets replaced with other minerals. So, you get the shape of it, and, there are things that get preserved and there are things that don't. So, yeah, it's a difficult, it's not very common to find things that are mostly soft tissue in the fossil record, but you can find them. So that's kind of an interesting.
Yes, and it does not take millions of years to make a fossil.
No.
It's been experimentally demonstrated that you can do it in about a week or so.
Yeah, it could be a week. It could be very, very quickly.
Yeah.
Yeah. So, one last thing I wanted to say about this was that you have, we also, so there are the bilateral embryos, or the early bilateral embryos, the early radial embryos, which are the symmetry. So you have something where you have a left side and a right side for bilateral embryos. The symmetrical embryos, you have this sort of circular symmetry. So you have like, it's almost like a pinwheel where you have different symmetrical parts. And then you have plant embryos. And plant embryos, I don't see plants on here, but it would be off to this side. Plant embryos are actually,
You got that.
well, I don't know if there are any good fossil embryos, but plants have embryos, just like animals, except that they're a little bit different. They're seeds. They have a development. They have cells that, there's an emergence from, cells and everything like that. And, we don't really know much about that. So, there's one of the papers I read, they said that there's they think it's like an independent evolution of embryos. So they think that like the embryo evolved multiple times in evolution, just based on the needs of this multicellular structure and the need to have like a complex morphology. But then other people suggested that there's a common origin of embryos somewhere, and that's unclear. It's unclear what that is.
Bradley, there's I can't remember the name of the plant. I think it's maybe some kind of, it's fucus. Fucus develops things that look like leaves, but in the middle, in the leaves, you'll find embryos being made. Can become new plants. Okay, so they're distributed across the whole leaf-like structure.
Yeah.
Okay. So they may not be just seeds.
Oh yeah. So these are embryos in the
leaf of fucus.
Oh, okay.
I'm pretty sure it's fucus.
Yeah.
The embryos have been studied a lot, but I can't recall any papers on the embryogenesis of it.
Yeah.
Or how should I say, not the embryogenesis, the initial formation of it.
Right. Yeah. All right. Who's Max Braverman? You had mentioned. Oh, this is. Oh, okay.
Put it in the chat.
Oh, fucus. Okay, like mucus, but fucus. Yeah. All right. Well, that's good. Thank you. Yeah. So that was yeah, that was a nice discussion. And so yeah, that's available. I put it in the chat. You can read it over. And talk about this more later. This is actually an image of if you're interested in stable diffusion and in generative machine learning models. I created this image from stable diffusion. I typed in Darwin as an embryo as a prompt, and it gave me this picture of Darwin kind of as like in an egg. His neotenous face, which means it's very young looking, but it's an old man. Yeah.
Are you saying Darwin was born old?
I guess, yeah. So that's interesting. So yeah, then I found this other article in the Nautilus. This is by Philip Ball. He's a science writer, and he said, if not Darwin, who? And this is an alternate history. If you're interested in alternative histories where, you suppose that history unfolded in a different way, then this is probably for you. So this is an alternative history of the great ideas of science. So he mentions Darwin, but he talks about a couple of different scientists here. Basically, the idea is if they hadn't existed, like if Einstein had never existed, what would physics look like? And so, the question is...
I just read a 1906 essay on Huxley.
Oh, yeah.
Darwin's bulldog.
Yeah.
And he sort of regretted having been much younger than Darwin because he figured he would have discovered evolution. Otherwise, Darwin didn't exist.
Yeah. Yeah. So this just kind of goes through this. So, it's really speculative, but basically, it's just kind of an interesting exercise in thinking about this. He said, these theories and discoveries probably would have materialized anyway sooner or later. So, it's nice to revere people, individual figures, but sometimes these things will come about one way or another. And so it's really about like us reading the literature and figuring things out, and someone's going to do it. And sometimes, it's just the person's in the right place at the right time. Sometimes that person really has skills that make that advance. Like I think Darwin going to the Galapagos was, obviously key to getting down the theory, because people had been talking about a lot of ideas in evolution and how that happened. But, and then you go to a place where you have a lot of tropical species, you have a lot of what they call adaptive radiation, meaning you have these different islands where different different species occur and have diverged based on their, isolation. Those sorts of things were necessary, I think. But still, people would have done this at some point. So this isn't like, didn't necessarily require Darwin. Yeah.
Huxley went on a trip around the world too when he studied jellyfish.
Yeah.
Okay. Might have led to it.
Yeah. So it's definitely a nice little exercise. And then this talks about the great man view of history, which means that you have a hagiography of one person versus like this process view of history where you look at the ideas and you say, those ideas were driven forward, and there may be different ideas that emerge, but that's basically the difference here. So he talks about heliocentrism and Johannes Kepler, and some of the things that happened previous to Kepler, like the Greek mathematician Arstiacus of Samos, and other people in medieval Germany and other things. So, there are differences in Copernican theory that are mathematical and, have its own flavor, but these ideas were around before. They were just, put forward in this way by this person. So talks about the laws of motion, Huygens, talks about James Clerk Maxwell and special relativity, which is not general relativity. It's a different, different way, a different theory. But again, this is stuff that was around. There were different things. There were different loose ends that had to be resolved. So in this case, you had an attempt. There was already an attempt to square electromagnetic theory with relative motion before Einstein, concocted by the Dutch physicist Lorentz. It too invoked contraction of space and time after a fashion, but did so with a view to preserving the ether, which was a concept they used before Einstein's space-time. So they had different concepts, and some of the concepts were wrong, but they were basically very similar to other concepts that become, that are actually right, maybe, or more predictive, we should say. So the ether is like space-time, except the ether didn't predict things very well, and space-time did. So we discarded the ether, and Einstein came up with general relativity, and space-time became the predominant idea. So this is,
Could you please put the URL in the chat?
Yeah. So this is this Nautilus article. All right.
Thanks.
All right, no problem. Now I'd like to talk about a group of papers that have to do with swarming behaviors and the behaviors of physics. So it could be chemical sensation or the behavior of agents and flows. And so the first paper we're going to talk about is this paper entitled Diverse Behaviors in Non-Uniform Chiral and Non-Chiral Swarmulators. So what's a swarmulator? The abstract reads, we study the emergent behaviors of a population of swarming coupled oscillators, dubbed swarmulators. So these are coupled oscillators, which are generally two agents that are synchronized or have some sort of oscillatory behavior. And so we have a population of these. If you're familiar with swarms, you would know that you could have a bunch of insects or a bunch of other types of organisms or agents that swarm around a common resource or form a common swarm. And it's just a body of collective behavior. And so we'll see examples of this later. Previous work considered the simplest idealized case, identical swarmulators with global coupling. So this is where everything is homogeneous and the coupling is global, so that means there's no variation within the swarm. Here we expand this work by adding more realistic features, such as local coupling, non-identical natural frequencies, and chirality. So the frequencies are shifted so that there's heterogeneity in the swarm. The coupling is local, so that means that there's heterogeneity in the swarm. And chirality simply means that the shape has a sort of handedness. You may think of chirality as like the orientation of DNA or of proteins where they're right-handed. So that means that they turn to the right when they move. And so this is something that you'll see in swarms as well. This more realistic model generates a variety of new behaviors, including lattices of vortices, beating clusters, and interacting phase waves. So these are dynamics that you'll usually see like in cardiac muscle, or you'll see this in what they call scroll waves or other things. And we'll see examples again later. Similar behaviors are found across natural and artificial microscale collective systems, including social slime molds, spermatozoa vortex arrays, and quink rollers. I'm not sure what quink rollers are actually, but these are just some examples. Our results indicate a wide range of future use cases, both to aid characterization and understanding of natural swarms, and to design complex interactions in collective systems from soft and active matter to microrobotics. So there's a lot of potential use for this in bio-inspired robotics, but also in soft and active matter, and then understanding biological swarms. So we have synchronization, which define as self-organization in time, and swarming, self-organization in space. So they have this self-organization, variable both in time and in space. And this is how they define swarms. So swarms can be synchronized. Swarms are actually the spatial component. Synchronization is the temporal component. So you see this sort of synchronization aspect occurring in flashing fireflies, firing heart cells, spiking neurons, and chorusing frogs. And it's basically where they synchronize their signals. Like in chorusing frogs, you'll hear like a din of frog song, as opposed to a bunch of independent frog songs all overlapping. You also see fireflies synchronize their flashes in response to different opportunities in the environment. Swarming, on the other hand, is seen in cell collectives that migrate in response to external signals. Flocks of birds that seamlessly change collective flight direction, and schools of fish that coalesce and move together to the collective's advantage. So these are all things that are spatially organized, and we can understand them as spatial phenomena. A combination of synchronization and swarming also occurs in diverse contexts, from biological microscale collectives and chemical micromotors to magnetic domain walls and robotic drones. So there's a lot here that you can look up in terms of the examples. Synchronization research has been, broadly speaking, focused on oscillators, which may synchronize in time, but not move around in space. On the other hand, swarming research has done the reverse. It has studied units moving through space that synchronize spatially dependent variables like orientation, but not internal phase variables in time. As such, the interplay of synchronization and swarming defines a new kind of collective dynamics about which little is known. So this is kind of the idea of the swarmulator. It's combining these two things and putting together a mathematical model to get us further down the road of understanding these. So swarmulators have a spatial position x of I and an internal phase, which evolve according to equations 1 and 2. So these are the equations they use to show the time evolution of these oscillators, these collective oscillators. So they consider swarmulators several different cases of natural frequencies, signal frequencies, two frequencies, single uniform distribution of swarmulators for their natural frequency, and then double uniform distributions, where half the swarmulators have their natural frequency randomly selected from a uniform distribution, and the second half have their natural frequency selected from another uniform distribution. So this is an example here, figure 1. This is the space phase order parameter. So they have 5 emergent behaviors that they used in the original swarmulator study, which we won't talk about, but they can replicate 2 of those 5 behaviors in this example. So you have A as the static synchronization, where the parameter S is near 1 or near 0, and then the static phase wave, which is B, which is where S is near 1. So you can see that between 0 and 1, there's the synchronization. You have variability here. You have this hole that forms. And so you have this difference in synchronization, and shows a difference in the swarm. And so there are ways that you can analyze this. So one of the things about a lot of collective behavior research is there hasn't been a lot of work done on sort of analyzing these shapes that form from collective behavior. So this is actually, I think, an opportunity for topological data analysis, but also other types of analysis that give us insights into these shapes. So they talk about non-chiral swarmulators. They talk about other types of behaviors, which leads us to figure 2, which is where non-chiral swarmulators, no natural frequencies spread. This is showing some of these phase diagram of different phases of swarmulators. So for example, you have this A example where you have disorder. So this is where there's no synchronization, there's no pattern formation in this swarm. You have the phase wave, which is this wave that goes across the collective, Then you have synchronization, which is here where everything is synchronized. And then you have synchronized expansion where the swarm expands, but it's in the same sort of form as this synchronized example. And this is the S parameter. So the S parameter is highest for the phase wave and lowest for the disorder, actually all of the other examples. This B is showing these different swarms in different ways of their phase waves here at the top. And this is a little bit different kind of behavior here where you start to get clustering within the swarm. And then you get this ring on the outer ring on the edge. And you know, bouncing clusters, which are clusters that bounce around. You get anti-phase behavior, which is where things are you have two different states that are anti-phase. So you have one set of oscillations peaking at one time and another set of oscillations peaking at another time. And that results in this sort of bipolar segregation within the swarm. Then finally you get radial oscillation, which is where you get oscillations that are radial. They go around in the circle and they take different forms. So this is, again, the S parameter. It's highest for phase waves and bouncing clusters and minimized for disorder, anti-phase, and radial oscillation. So this is a phase diagram. Figure 3 are the order parameters for non-chiral swarm relators. This is for disorder. So you have active phase waves, splintered phase waves, and synchronization at the very top for this K parameter. Radial oscillations show different pattern. Disorder actually shows a different pattern this is for the J parameter. This is for the K parameter. J equals 1, K equals 1. This is J equals 1 for disorder. And then J equals -1 for disorder. You can see that as you change these parameters, they change as well. The results change for these different things. All right, that's a little bit harder to unpack if you haven't read the paper, but this is actually a little bit better example of what's going on here too. You have, again, these phase diagrams of the S parameter. So the color gradients are the S parameter changes. And then these are the examples of the swarms here. So you can see that you have all sorts of different behavior for swarms. You get these vortices, which are where the agents circle around a central point. You get dense revolving clusters where they revolve around very dense packings. You get sparse revolving clusters, so they revolve around a central point, but they're much less densely packed. And then you get vortices, which are where you get these circular movements. So these are very similar things, but they exhibit different, they inhabit different parts of the phase space. So for the S parameter, it's lower for these kind of clusters here. For these kind of vortices, the S parameter is higher. For dense revolving clusters, some of them have a high S parameter value, and then for some dense revolving clusters, transitioning to sparse revolving clusters, the S parameter is lower. So you can see that the time evolution and the time evolution is connected to the spatial evolution, and it has a lot of interesting dynamics. In this case, you even have an even more complicated phase portrait. You have these double revolving clusters where you get two centers of mass, and you get clusters revolving around those points. You get sparse oscillating clusters where things oscillate around, but the population is much sparser. And then you get these vortices again for high values of the S parameter, and then for low values of the S parameter. Those data aren't shown, but they would be a little bit different probably. But the point being is that there's a lot of variation in the phase portrait. And so they've been looking at the global level so far. They've been looking at the order parameters, and changes in the order parameters exhibit changes globally in those structures. And so we can see, again, that you can change the order parameters, and you can find, this is actually figure 6. These are frequency-coupled chiral swarm relators. So these are, again, phase portraits where you get these different types of behaviors, and depending on the S value, you can classify different parts of this phase. Actually, it's the S parameter and then the J parameter and the K parameter. So this is K versus J, and that tells you your sort of position in phase space, and then the S parameter is the color. So again, we have these different types of patterns: single phase wave, double phase wave. The double phase wave is where you have two centers of mass. The single phase wave is where you have a single center of mass and where they oscillate around that center. And then you have these different oscillating clusters. So you have these dense clusters and these sparse clusters. And then for this one here, it's even more complicated. You have these double vortices that emerge at high levels of J, for moderate levels of K. You get these double clusters for high levels of K and most levels of J. And again, most of these examples are with a relatively low to moderate S parameter value. Same for double vortex. Single vortices, however, have a much higher S parameter value, and that's for low values of J and moderate values of K. So you get this kind of single vortex, and then you get the this type of disorder, which is just nothing that makes any coherent sense, and that's at very low values of J and K and very low values of the S parameter. So you can see all that variation over time. And this is an interesting way to analyze swarms. Again, there are no really good methods for doing this. This is probably the best method I've seen, or one of the better methods I've seen for doing this, mapping this to a phase space and then interpreting the clusters, different patterns that get formed. But there are probably even better ways to do that, than that. But I have not seen good papers on this. So that's that paper. And actually, one more thing I will show here is that you have these correlation functions. So you have these phase-phase correlation functions where you show different patterns in the swarm and these different values here in these graphs. So if you want to read this paper, I would go ahead and dive into it. Now this paper is on Jeffreys orbits and microswimmers and flows, and this is a theoretical review, so this is going to be a pretty dense paper. I'm going to go over the abstract and then maybe through some of the math, but probably not too much of it. So in this review, we provide a theoretical introduction to Jeffreys equations for the orientation dynamics of axisymmetric objects in a flow at low Reynolds number, and review recent theoretical extensions and applications to the motion of self-propelled particles, so-called microswimmers, and external flows. So a Reynolds number is a number for resistance. So if you're in a flow, you have a resistance to that flow. If you're a cube and you're traveling through the flow, you have a very high resistance. If you're a sphere, you may have a lower resistance. Similarly, if you have a highly packed, like a capillary filled with a flow, and you're going with the flow, and there are a lot of you, the Reynolds number will be higher. If there are fewer of you, the Reynolds number will be lower. If you're in a turbulent flow as opposed to a laminar flow, that Reynolds number will also be higher. So it depends on a number of things. It depends on the turbulence of the flow, it depends on the shape of the body, and it depends on the density. So these are all things that we have to think about when we think about flows and microswimmers. And so the Jeffrey equations, a simple set of ordinary differential equations, provide a useful building block in modeling, analyzing, and understanding these dynamics in a flow current. And so particularly when incorporating the impact of the swimmer's shape, since the equations contain a shape parameter as a single scalar. The particle orientation forms a closed orbit when situated in a simple shear, and this non-uniform periodic rotational motion, referred to as Jeffreys orbits, is due to a constant of motion in the nonlinear equation. So it's a Jeffreys orbit is a non-uniform periodic rotational motion. So this kind of relates to the last paper in that you have this a lot of analyses give this assumption of of homogeneity, and being able to analyze these kind of moving systems, whether they be flows, whether they be swarms. It's useful to have a method that really can capture a lot of the heterogeneity that exists in the real world. So they provide a theoretical introduction to microswimmer hydrodynamics, and then derive the Jeffrey equations, and then they extend this to more general shapes, including those with rapid deformation. So if we think about something like an organism has, maybe a different shape. Maybe the shape changes over time, maybe over development. You go from a spherical shape or an oblong shape to something that's asymmetric. And even if you remain like a single cell organism, you can still experience deformations. Sometimes those are due to stresses and strains of the environment. Sometimes those are due to the membrane, the rigidity of the membrane. There are a lot of things that play a role, but again, this is going to tell us something about these swimming dynamics. If you have more deformations, if you have a higher Reynolds number in your world, if your world's a higher Reynolds number world, you may need to adapt to that. And so that's what we're dealing with here. In the latter part of this review, simple mathematical models of microswimmers and different types of flow fields are described with a focus on constants of motion and their relation to periodic motions in phase space, together with a breakdown of degenerate orbits, which are the degenerate orbits of the dynamics, to discuss the stable, unstable, and chaotic dynamics of the system. So again, we have this phase space where we have the chaotic, the unstable, and the stable, the unstable, and the chaotic dynamics. And so this is something that can be extended to laboratory experiments and simulation. So they go through some of the microswimmer hydrodynamic governing equations. We use the Navier-Stokes equations as inspiration here. Then we have the equations of motion in a swimmer immersed in a fluid. We have the motion of a deforming object. We introduce two reference frames, a laboratory fixed frame and the swimmer fixed frame. So you have the participant and the observer. The linear and angular momentum conservation should hold. So at low Reynolds numbers, all inertia terms are negligible, which is always usually the case. But at higher Reynolds numbers, you have to account for inertial motion. And so we go through this. We go through the flow that satisfies the Stokes equations. The total fluid velocity is the sum of the background flow and the distributed flow due to the presence of swimmer U sub d. That is U equals this. In many situations in nature in the laboratory, the length scale for modulation of the background flow is larger than the size of the swimmer. So we're dealing with very small systems. We're dealing with very small swimmers, and they're not at that sort of nanoscale barrier, because when you get down to the nanoscale, maybe less than 10 microns or so, or maybe less than 100 microns, you start to get this effect of where you get too small to really be governed by the Reynolds number. There isn't enough, the effects of turbulence aren't as great. The effects of the swimmer's body isn't as great. So those equations break down around that size limit. So we're above that size limit, but we're still pretty small. So we're smaller than, most multi-cell organisms. I guess maybe some multi-cell organisms are that small, but you get my point. So even in the turbulent ocean, the smallest length scale of the vortices are estimated at around 10 to 30 millimeters, which is sufficiently large to approximate the local background flow to be linear in position. So we see that, sometimes the length scale is smaller than some of these vortices that we're dealing with. Much smaller, in fact. So we actually go through these equations again. We introduce a Lagrangian. Then we focus on the kinematic problem, one which solves the swimming velocity u. And then thus the swimming trajectory under a given shape gate. So that means how the shape moves through the environment. If we need to solve the shape gate as an unknown function by solving a coupling problem with the material elasticity, this is called elastrohydrodynamics. And so we can combine different things. We can combine hydrodynamics with material elasticity. As I said before, we have to account for deformation in the shape. So we can see that this is the swimmer. The swimmer might deform significantly in the flow, or it might not. We have to account for those with our equations. And we can, of course, can do this in our modeling. It's just a matter of what we want to assume. So then we have a lot of other cool math. There's a swimming formula, and this is an example of a slightly deforming microswimmer. So this is a single cell organism, I believe. I don't know what it's supposed to be. It's just a generic organism with cilia, but you have these deformation points on the surface where things get deformed, and of course that affects the Reynolds number and affects how it interacts with the flow and all of this. So there's a specific type of swimmer known as the squirmer. The squirmer was first proposed by Lighthill, and it is a slightly deforming sphere later applied by Blake to analyze a swimming cilia. Now they're talking about a swimmer referring to a mathematical model of a rigid body with a surface slip velocity instead of an active deformation. So we can actually approximate the deformation by using surface slip velocity. Due to the model's simplicity, the squirmer has been extensively studied in the past few decades to elucidate universal features of microswimmers and their hydrodynamic interactions, as well as to model the behaviors of specific microorganisms, such as ciliates and volvox. And actually that's what this kind of looks like, is volvox. If you look at this, where you have all these spheres inside of this cell, that's what volvox looks like, but you also have cilia on the edge. So it's kind of an amalgam of these ciliated microorganisms and volvox. And of course, artificial swimming colloids, which are another thing that we can, that are very similar to that. So there's a lot of math here, and then you get into the Jeffreys equation, and you get into some other things of looking at the effects of shear on this microswimmer, or at least on a sphere, and then you can use that to understand what's going on in the microswimmer. You have hydrodynamic symmetry, so you have these other things you have to consider, and so you can see the parallels between this paper and the last paper. There are theoretical extensions for more general shapes, as promised. You can look at helices and helicoid objects. You can look at corkscrews, which are movements of bacterial flagellum. So the flagellum moves in a corkscrew motion to move the bacteria. This is, of course, going to affect the surrounding medium, the surrounding liquid medium, and create a wake and create inertia. So these are things that you can model as well. And a simple helix, however, does not rigorously satisfy the definition of helicoid symmetry unless the number of turns is an integer. So there are a lot of mathematical considerations you need to consider here. There are also different types of shape parameters for bacterial cells, swimming ciliates. So these numbers exist in the literature, and you can just plug them into the model and test your model and see how it behaves in reality. So this is an example of the spermatozoa and the swimming trajectory of a sperm head. And, this is something that is sort of driven by Brownian motion. So, we can track that and look at its dynamics, but it also interacts with its liquid environment. And so you can introduce shear to this sort of microswimmer and see the effects. E being the components of shear, and then V being the forward velocity, and then the angular velocity here. So this actually shows the drift torque and the drift force on this body. This is a helicoid object, which is the helicoid tail under a simple shear, force. So there's drift force in this direction, drift torque as it turns, and those are the parameters you need to model. So I'm going to go to the last paper here, in the interest of time. And this paper has to do with the chemoreception and chemotaxis of a three-sphere swimmer. And so this is where we're considering chemoreception and chemotaxis. So chemoreception is where you're taking in chemical information into an organism. It could be a nervous system, it could be a chemical signaling pathway. And then chemotaxis is where it's moving towards a chemical gradient or along a chemical gradient. And we're dealing with this three-sphere swimmer, so it's a segmented spherical swimmer. So in the last paper, we talked about a lot of these types of models, the hydrodynamics, the Reynolds number, and then there's also the Piclet number, which is another parameter that we need to consider. This is a numerical simulation of the problem with a finite element code based on the Phoenix library. So this is using a finite element approach. For the swimmer executing the optimal locomotion gait, we report the Sherwood number as a function of P in homogeneous fluids and confirm that little gain in solute flux is achieved by swimming unless P is significantly larger than 10. So then we also consider the swimmer as a learning agent moving inside a fluid that has a concentration gradient. The outcomes of Q learning processes show that learning locomotion with the displacement as reward is significantly easier than learning chemotaxis with the increase of solute flux as reward. So this is interesting. What they're doing here is they're taking the swimmer and they're looking at the hydrodynamics of it. But then they're also considering the swimmer as a learner of chemical information. So it's learning about the chemical gradient and it's producing an outcome or like a taxis. And so they're able to use Q learning, which is something that they use in reinforcement learning and other related techniques. It's a process, so it's very analogous to reinforcement learning. It's actually, I think, just a form of reinforcement learning, but it's not quite the same. Learning locomotion is something that you can do with a reward. So you learn based on repeated rewards. So you can move the learner or move the swimmer in a certain direction behaviorally by having a reward that is sort of iterative, that you can acquire a reward at every step, and eventually you learn this is the correct behavior. So we can use displacement or we can use solute flux. And it turns out that it's easier to use displacement than solute flux. The chemotaxis problem, even at low PE, and this is the Piclet number, has a varying environment that renders learning more difficult. Further, the learning difficulty increases severely with the Piclet number. The results demonstrate the challenges that natural and artificial swimmers need to overcome to migrate efficiently when exposed to chemical inhomogeneities. So the Piclet number has to do with chemical gradients, and the Reynolds number has to do with the hydrodynamics. And so we need to, again, understand these homogeneities and heterogeneities. And in chemical homogeneities, we often see these in chemical gradients. Chemical gradients aren't necessarily linear, but they need to learn how to migrate according to these signals and according to the hydrodynamics. So this is, looking at these type of things. There are a lot of, uses for understanding this, understanding these small, systems. It's important in embryo development and in immune responses and in understanding things like algal blooms and cell migration and the microbial life cycle. So the swimming strategies employed by natural and synthetic microorganisms are highly diverse. To gain understanding of the basic mechanisms, theoretical studies have frequently relied on simplified models. And then, of course, again, they bring up the squirmer, which is this model of spheroid particles with a given split velocity over its surface. This was introduced by Whitehill and Blake, and they also cite Stone and Samuel. Another simple early model is the Taylor sheet, and this is by Childress, a flexible sheet that swims by deforming transversely according to a moving wave. And so they address, in this study, a three-sphere swimmer model introduced by Najafi and Golstanian, inspired by earlier three-link mechanisms proposed by Purcell. So this is where you have a swimmer with three aligned spheres with two contractible arms of 0 thickness joining them. It exhibits quite large surface motions because of the variable arm length with the simplicity of one-dimensional kinematics and spherical constituents. So let's see if I can find a picture of this swimmer. So this is basically it, the three-sphere swimmer. You have, this is from Najafi and Golstanian. So you can see that you have three spheres. They're joined by these two rods, and they're variable length. So you have L1T and L2T. These are different lengths between the different spheres. So the first sphere is way out in the front. The other two spheres are towards the back. And so this has an effect on the swimming. And this has an effect on the hydrodynamics. But it also may have an effect on how it learns from a chemical gradient, because you have spheres in different positions. There's a leading edge and a trailing edge. And so this gives you some flexibility in how hydrodynamics and unlearning may occur. And so, assuming no external forces acting on the swimmer, the hydrodynamic problem reads, given x0, L1T, and L2T for t or times larger than 0, find xt, uxt, and pxt such that, subject to these constraints, the gradient of the gradient. So what we're trying to do is we're trying to find the optimal solution in this gradient. And so equations 2 and 3 are the well-known Stokes equations. Is the force-free condition where n is the unit normal outward and imposes the no-slip condition at the surface of each sphere, being the unit positive direction of motion. Coupled to this hydrodynamic problem is the solute transport problem, in which the unknown is the concentration field CXT. So given the initial concentration field for times larger than 0, find CXT satisfying these factors. So this is again the gradient. We're looking at the boundary conditions that are far away from the swimmer, so we need to optimize for that. And so you deal with the solute, and the diffusivity of the solute. And so this is the moving gradient, basically. So the chemical gradient diffuses over time. We need to account for that. And so we, I think that's enough for this paper. I think that this basically is an application of what we talked about in the last paper, and in the first paper, maybe a little bit more indirectly. So I think I'll finish up with this figure. This is the swimming gait for three-sphere swimmers. The sequence is from top to bottom and follows enumeration introduced in section 5.1 for states and actions. So they're actually looking at how this thing is doing using Q-learning to learn. But this is basically the configuration here. S equals 4, S equals 2, and then A equals 1, A equals 2. You'll have to read the paper to understand that a little bit better, how they have to set up. But basically you can see that the shape of the swimmers is a little bit different for each of these differences in parameter for states and actions. So you can see that you have a state here, and then you have a different action. So, yeah. And this finally gets into the Q-learning, and they talk about the reinforcement learning framework and some of the things that they're. So we define that the agent must move exactly one arm in each time interval from the fully extended position to the fully contracted position, or vice versa. The only decision to be taken at each discrete instant, t, is whether to move arm 1, action 1, or arm 2, action 2. At each of the 4 possible states, there are just 2 possible actions. This kinematic restriction on the agent rules out the possibility of the swimmer remaining still in any position. The swimming gait to the right, starting at state 4, corresponds to the sequence of actions. And so the agent can be made to follow a policy or a rule that each state S associates a probability of taking action at time t. So you can see again, actually we'll go to figure 5 and show the changes in the swimming gait based on this learning process. So the gait changes as the learning process proceeds. You get these, this is a map of state transitions here on the right, and on the left you have the the swimmer spheres in a vortex, how they behave there. So that's all I wanted to talk about. All right. I think that's. Do we have any other things we wanted to talk about today, or?
Bradly, I want to mention one thing.
Okay.
After this proposal, I will start working on that model in Hugging page to host our models. So it's like getting kind of late.
Yeah.
I will do that work after the proposal work completes, because the proposal deadline is coming, right? After that, I will do.
Yeah. Yeah. So yeah, I think that's great. Definitely work on the proposal first. That should be your first priority, and then you can work on this other stuff. So word about that. The GSoC proposal is open on March 20th. So that's coming up, but that's just the beginning of that period. There's a deadline of, I think, in April where the submissions have to be made. What was that?
April 4th, Bradly. April 4th was the deadline.
April 4th, okay. So the road to this deadline is if you're interested in developing a proposal, Sushmanth has already gotten his draft together and it looks good so far. You're going to have to probably make some iterations of it where you have to you want to let me see it before you submit it, definitely. And I can give you pointers on how we can refine it. And so I would do that as soon as possible. I wouldn't wait until March 20th. But what people tend to do is they tend to have this period where they're interacting with their mentor, which would be me. And I encourage you to interact and, maybe make some contributions before the deadline. So if you're interested in, if you want to explore the org, that's fine. I can send you things. We usually do this in the Slack. So if you're in the DevoWorm channel in the Slack, that's good. We also have a DevoLearn channel, DevoLearn DevoGraph channel, which is also somewhere where we usually post a lot on DevoLearn and DevoGraph. So that you'll want to join that channel as well. And so when we get closer to the deadline, I'll be sending out materials, maybe more on some of these topics. If you have things that you need me to explain, I can explain them. And then, there's a specific portal to submit work. So your proposal will be submitted through a specific portal that Google has set up for this. And it's just basically where, there's a certain format for the proposals. It's basically where you have an introduction to yourself, and then you describe your approach to the problem. So you lay out what you're going to do and how, if it works or not. Sometimes that'll require you to do a little demo and put screenshots in the proposal. And sometimes it just requires you to be very clear on the outline. Like, what is this thing I want to do? How does it relate to what has been done in the org? How can this contribute to solving the problem? And in our project here, we're doing a lot of sort of maintenance. So, I mean, the way it's written is that there's all this stuff in our. That's why I showed you the repositories. There's other stuff in our repositories, and this project is kind of in part to kind of go through there and maybe make that better, or to drive that forward in some way that is not starting a whole new project necessarily, but just to improve upon what's already there. So that's really where you want to go. And I think Sushmanth is introduced, I think, has a pretty good start on this. But if you want to do this, if you want to be really, I think, have a really unique proposal, I would go through all the materials and see what we have, and kind of describe how you might tie this into some of the other organizational things. So like we're doing image segmentation, and then of course we have the graph neural network stuff, but we also have the lineage tree stuff, and we have other types of things that we've done in the past, both within GSoC And then in the DevoWorm GitHub repositories, we also have things that aren't GSoC related. And those may or may not be relevant, but they may tie into those. And then you'll need a schedule, which is where you have to lay things out week by week. And so laying things out week by week is typically, it's not as easy as it seems. You may want to, kind of think about this as like a schedule where you go through your work, but you have a contingency plan. And so the contingency plan is where if you're, you have this path to success, right? You want to do all these different things and steps, but what if something doesn't happen the way you want it to? What if something doesn't implement correctly? Or what if it takes you a while to implement something? You'll want to sort of try to implement things very early, maybe even during your proposal period. But, sometimes you don't know when you get into something. We've had problems with like people running out of computational resources or having problems with bugs. And that's fine. That's perfectly fine. That's what the program is about. You want to be able to overcome those. So in your proposal schedule, you want to have like your schedule, the things that are implemented step by step. And then you want to make sure that you have things in parallel so that if you get stuck on something, you can go to something else, some secondary task. So if you're trying to implement a toolbox, for example, and the toolbox isn't implementing, you can stop doing that for a while, move to something else, and then try to solve that toolbox problem. And then once you get that fixed, then you can implement that. But you haven't wasted a bunch of time because it's a very tight schedule. It's like, 12 weeks or something. So you can't really take time off to figure it out. And that's one of the challenges of GSoC is that they're trying to teach people how to, work within a very tight schedule and get things done. And so that's what my advice to people. I also suggest that, if you're, you don't have to stick exactly to that schedule. We can rescope the project, but it's good to have it pretty close to what you're going to do, because rescoping it is a problem. You can't get as much done, and sometimes it's, hard to really rescope something if you're halfway down the road. So I would suggest really kind of thinking through what you want to do and making sure it'll work, or being pretty sure it'll work before you propose it. So.
How many pages proposal should be there? Like, mine is going around 17 pages. Like, there is any ideal number?
Yeah, I think probably. I've had people in the past, maybe 8 to 10, because you're going to have like references. You're going to have your schedule. You're going to have your stuff that your images. I think images are all the successful ones I've had, for the most part, have had images, just where people will try out something. They'll write a little notebook and they'll have screenshots of whatever, and it just shows like how something works. And, maybe it's 8 to 10 pages, maybe longer. But it's really about getting the argument in there and saying, this is what I want to do. That's pretty much it.
Is there any prior application process which, like an application we have to fill during this time, the contribution period?
No, we don't really have, there are no pre-applications. It's just, if you can make like some pull requests during that application period, that's good. It'll maybe help you think through some of the ways that you can contribute and maybe ways some of these things work. But it's up to you. Really, the only thing you really need to do is do the application and get that done. And that's really the most important thing, because that's what we'll be evaluating. All right. Well, thanks for attending. And again, we'll be discussing some of these things in Slack. It looks like Dick put something in the chat. This is, oh, you're muted, Dick. Oh, this is a whole bunch of references. I think he came up with some references here. There we go.
Yeah, these are all my references on fossil embryos.
Oh, good. Yeah.
The fellow I was thinking of was Bankston.
Yeah, there we go. I see it.
Okay.
Great. Yeah.
So he did three-dimensional imaging using X-rays. Okay. So somewhere in these papers that data might be referred to.
Yeah, that would be great to see if we could get some of that.
I had to go to my other computer to get it.
Oh yeah, I saw. Yeah. Well, thanks. Yeah, that looks good. Yeah. Well, that's all for today.
Okay.
Thanks. Have a good week.
Bradley, I shared the presentation, whatever the presentation I have done.
Okay.
I shared in the chat.
Yeah, that sounds
You can
good.
go through, like, if you have any doubts. We can do.
Okay, yeah, sounds good.
I also wanted to ask about the Origin of Life project you were working on, and you talked about it last meeting.
Yeah.
Yeah. What's your question?
I was just curious. I could not find any papers or some resources.
Oh, there are 70,000 papers on the origin of life.
Like, do you want to work on?
I have not been able to read them all.
Yeah.
Okay. I'm taking a different approach. Are you familiar with oil and water emulsions, or what's called Italian dressing? I don't know if you have Italian dressing in India. Do you?
We do with Italian food, I guess.
Okay. Okay. If you shake a bottle of Italian dressing, the oil disperses into little drops. Okay, and you can see them. It's called an emulsion. They last for a little while until the drops go to the surface and break. Now, if you take something like that and you cool it slowly, like over a period of a couple of minutes, those drops become flat and they become polygons. Okay, and this was demonstrated by civil engineers back in 2014. Okay, so it's a brand new, it's a new result. And when I saw it, I've been casually aware of what are called archaea.
There we go.
Okay. Archaea are prokaryotes. They look like bacteria, but they're very different from bacteria biochemically. Well, some of them have flat polygons. So they may go back to the origin of life. So I'm working on the hypothesis that origin of life was a transition from what are called shaped droplets, polygonal droplets that are flat, to polygonal archaea. Okay, so that's what I'm playing with.
Okay.
Okay, send me your email. I can send you some papers.
I'll just write it on the chat.
Okay.
Okay. And the advantage of this is it gives a starting and an end point to the origin of life, which no other theories do, because they all assume that somehow cells become magically nice round cells.
Yeah.
And a lot of people argue whether the cells came first or last. Okay, got it. Okay, Kritika, I'll send you some papers on it.
Okay, thank you.
I know it's late in India, but here I haven't had breakfast yet.
Pardon?
I know it's late in India, but I have not had breakfast yet.
Yeah, it's actually almost 10.
Yeah.
Yes, I know. I know. Here it's a little after 10 a.m.
Yeah.
Okay. Okay. Okay, got it.
All right.
Okay. Anyway, Bradley and I are working on one paper on this subject.
Yeah.
Right.
Yeah. The paper we saw last week, and yeah, we'll see how that goes.
Yeah, I got to make sure I didn't screw up his equations because they were not coming out correct.
Ah.
So I took out the equations had little boxes in them, which are not symbols. So I took those things out. And I had to make sure it did it right.
Yeah.
Okay.
All right. Yeah. All right. Have a good week,
Okay,
everyone.
have a good
Thanks.
week.
Take care.
Bye.